Bradley Burcar. I'm a NASA postdoctoral fellow at Georgia Tech. I work at the Center for Chemical Evolution, where our main focus is to look at basic prebiotic chemical systems and see how those can grow and evolve and develop complexity over time. Because if you think about it, if you look at biology and you really tunnel down to its basics, it's all just chemistry at its heart. So as a prebiotic chemist, looking at the origins of life, I love to think about this era of Earth. Back when you have a primordial soup, perhaps, maybe you have hydrothermal vents, maybe you have these small little warm little ponds and lagoons on the surface of the Earth, where all sorts of interesting and chemical complexity can happen. So eventually, the complexity grows. You develop membranes, RNA, DNA, proteins, all of the fundamental parts of life, and eventually transition from this sort of system to a living system of modern Earth full of life. But you have to go back to the basic fundamentals and figure out how those systems came to be in the first place. So my current project is focusing on trying to synthesize a very basic monomer. So for this case, you look at RNA or perhaps DNA. And it's possible that this was not the very first informational carrying molecule. But its components are quite informative on what the first informational bearing molecule could have been. So we don't necessarily, at the CC, think that this is the first monomer. But you can look at the bases and look at that as information carrying unit. And you can replace with something else, like Matt's talk earlier today with barbituric acid or other sorts of derivatives. But the main thing is something that does hydrogen bonding to carry information. You can also look at ribose, which is just a component that can tie all of the different parts of the monomer together into one system. It doesn't have to be ribose. Maybe it could be glycerol. Maybe it could be something more complex. Glucose, who knows? But as long as it can branch the different parts of the system together, that's the important part, as a trifunctional connector. And my specific part that I'm working on in this project is looking at this ionized linker. And modern life uses phosphate as its source. It makes very energetically favorable bonds that can be formed and broken quite reasonably. And it provides a basic amount of solubility in water. Very valuable properties to have for life as we know it, for polymers and monomers that have to exist in an aqueous environment. So it's not essential that phosphate was the source. And in my group, we look at a lot of different possibilities for what it could be for phosphate. But phosphate is so helpful, it has such good properties that it would be really great if it could be the source. And so I'm specifically trying to find reactions that could help to incorporate phosphate into early organics. But this has been a problem for a long time, the prebiotic phosphorylation problem, which is why people are even looking for other informational linkers as it is. Aha, it did it. Yep. All right. So the basic fundamental question is how phosphorylated nucleosides or organic phosphates came to be in the first place. There's two fundamental problems. First of all, if you have phosphate, it reacts with divalent cations like magnesium or calcium in solution, and it just precipitates out, and you have a nice bed of apatite. So this makes it largely inaccessible for prebiotic chemistry, which is the main reason why a lot of prebiotic chemists have dismissed it for so long. Also, if you're making organic phosphates, it's a condensation reaction. It gives off water. It is incredibly thermodynamically unfavorable to try to give off water for a chemical reaction in a water-based environment. So these are two big problems trying to make phosphate be incorporated into organics. So a lot of research has been done starting in the late 60s to figure out what can work. How can you take a nucleobase or any organic, incorporate phosphate, and make organic phosphates? So a lot of work has shown that urea is a very key component for this. If you have urea and soluble phosphate, like sodium phosphate in solution, in a dry reaction, it can add phosphate as low as 65 degrees. Or if you really ratchet up the temperatures to 100 degrees, you can take insoluble minerals like apatite and also phosphorylate. Also, if you just remove water completely from the equation and use something like toluene or formamide, you can do low temperature reactions also using soluble phosphate as low as 70 degrees. But it's kind of a cheat because you removed water from the equation, and then you have all sorts of arguments to make about formamide on a prebiotic earth. Also, they have pre-treated some of the insoluble minerals and have used that as a phosphate source to phosphorylate. So that's one way to deal with the insoluble mineral problem. But this pre-treatment took place at 150 degrees Celsius, which is very high temperature. And recently, other novel solvent systems have been explored, particularly this choline chloride urea eutectic solution. So this is also a water-free solution. And eutectics are interesting multi-component systems in which you can mix them together and you have a lower melting point than either of the components. So in this case, you take two solids at room temperature, mix them together, and you create a liquid. This is actually quite abundant in plants and a lot of biological life, so it has been investigated a lot in modern biochemistry. And if you use one of these interesting novel solvents, it's been shown that you can phosphorylate as low as 65 degrees. It's problematic because it reacts with your solvent in this case, which is not great, but it works. And I'll reiterate this part here because this is apparently a very important fundamental reaction in prebiotic chemistry. So this is the urea catalyzed phosphorylation. So you can take your phosphate group, take your urea, and create this high energy intermediate in a water-free solution. So in fact, you're activating the phosphate. If you can create this, then you can bring in any alcohol that we've tested so far and create an organophosphate. So it's a great way, and urea is a very powerful chemical for doing this. And the biologists in the group might notice that this looks like a EDC, activated intermediate, which is used for a ton of modern biochemical reactions. So we've been investigating if phosphorylation can take place in a eutectic made of urea, ammonium formate, and water. Well, this last part is key because it allows this to be created on a prebiotic earth in a water-based soluble system. So the great things about this eutectic, all of these components are very abundant on a prebiotic earth. We saw earlier the serpentinization reactions can lead to a lot of ammonium formate, for example, and urea can be abundantly made from Miller-Urey type reactions. In fact, a lot of prebiotic scientists think that like the salt flats in Utah, you would have had urea flats on a prebiotic earth. So both of these would have been very available. Also, after some slight heating, driving off the excess volatiles from the solution, you have a fluidic environment that can promote chemical interactions. So you don't have to have a completely dry system anymore, and a lot of chemistry can take place a lot more readily in fluidic environments. It's made from urea. Also, upon heating, these chemicals actually do form formamide, which creates a 4-part system. And then we have this solvent, which has been shown to do phosphorylation before, and some other prebiotic reactions. So it's a great, easy pathway to developing formamide. The high viscosity environment promotes chemical interactions. And it's a low enough water environment that it can actually still do dehydration and condensation reactions. And one of the first things that we discovered through our work with our collaborators at USF is just by taking different phosphate sources. Struvite, which is a mildly soluble phosphate source, hydroxyapatite, which is calcium phosphate, which is a very insoluble phosphate source, and vivianite, which is an iron phosphate, which is also quite insoluble, and mixing it in the eutectic, comparing it at 70 degrees to water, we could see that just initially in the eutectic, there's a marked increase in solubility of these highly insoluble phosphate species. So one good check for the eutectic to start with is great. You can now solubilize some phosphate. Also, thermodynamically, we ran some calculations and showed in a urea-rich environment that's slightly acidic, like the Eutectic is, if you have magnesium and sulfate in there with this insoluble phosphate source, you can just generate struvite, a much more soluble mineral, at a very thermodynamically favorable rate. So you can just mix it with the Eutectic and it just can pull the phosphate right into solution. And this is shown, we've ran some XRD and some Robin, actually testing this experimentally on some hydroxyapatite. And it's invisible, but there's a red line down here that's for the hydroxyapatite, which is, this didn't come out at all. Anyway, what this shows is that it works. You mix it together and you create struvite. You can separate all the minerals that you want and see that it has worked greatly for transferring hydroxyapatite to other sources. Oh, Max. So this allows us to create a very fundamental prebiotic model. So you could take calcium, magnesium, and phosphate on a prebiotic earth, which come together and form this bed of insoluble minerals. The water can be removed, and then you can get a nearby hydrothermal source, some volcanoes or other surface vents, that can create this suite of organic molecules and inorganics to create the beds of urea and ammonium formate. Water will come, wash them into solution. You now have a nice warm lagoon rich in magnesium sulfate, urea, ammonium formate, which will then convert all of these minerals to the much more usable struvite and soluble phosphate. I'll go through my reaction conditions very quickly. It's very simple. You mix some adenosine with the eutectic in a phosphate source and heat it. And those are the reactions that go forth and make phosphorylation. So this is an HPLC trace showing the different phosphorylated species that are made. 5' AMP, some cyclics, 2' and 3'. And these chromatograms down here compare reactions with soluble phosphate in the eutectic to soluble phosphate in just a urea solution alone. So you can see the area of the peak is important. It's related to how much you have in solution. So the eutectic works. It makes great phosphorylated products. Comparing it to the urea controls, which are up here, we get a total of 7% phosphorylation at 65 degrees, but 58% running into the eutectic. At a higher temperature of 85 degrees, we just see a mild increase. So this works really great at lower temperatures. When you're comparing the different phosphate sources that you have, we get up to 32% phosphorylation with sodium phosphate. Struvite and euboreate, which are very similar minerals of mildly phosphate solubility, give a little bit decrease, but still a great amount. And then we synthesized some struvite ourselves in the lab, and it's a mixture of brucite and struvite and other mixtures, and it still works great. It's a very dirty mixture that still produces a decent amount of phosphorylation. And we processed some hydroxyapatite showing that model that I had before with the washing of the magnesium sulfate and the drying and converting, and showed an increase in phosphorylation compared to just hydroxyapatite by itself. And this is really emphasized when we do some wet-dry cycles, where we continually add in magnesium sulfate, dry it, add it in more to really drive the equilibrium forward. So this is particularly noticeable at 85 degrees when we get a decent amount of phosphorylation with sodium phosphate and struvite, hardly any with hydroxyapatite at 65 and 85, and it really jumps up and we get 15% phosphorylation by treating these mixtures with the magnesium sulfate. So we can follow that one pot scenario to generate that lake and solubilize phosphate and bring it into reactions. And also, it doesn't just work with adenosine and nucleosides. We have done this with glycerol. So we get phosphoglycerol. This is a phosphodiester bond of 2 glycerols and phosphate, and 2 phosphates and 2 glycerols. So this is showing the mass spec. So we can create higher order phosphorylated species and create phosphodiester bonds. If you're really paying attention a couple talks ago, you could think that this might work with fatty acids, and maybe you can make some lipids and some protocells. So all sorts of interesting possibilities. So overall, these results have shown that the Eutectic liberates the sequestered phosphate, promotes phosphorylation, and for the future work, what other reactions will work? We're taking this off in different angles and seeing maybe this is a great solvent to make all sorts of different prebiotic chemistry. Can it make long-chain polymerization reactions? We saw the 2 glycerols and the 2 phosphates, so that made a polymer there. We've seen up to 3 and 4 mers with the adenosine. Maybe we can make 10 mers, 20 mers. Maybe there's some magical chemical equilibrium that can happen in there to make the information carrying polymers that we need. And this paper has just been accepted for publication on Gavante Chemiae. So you can see a more detailed version of our work if you check out that journal in the next couple months. Here's the group that I work with and my funding sources from the NASA astrobiology program. I'm supported by the NASA postdoctoral program, and we are funded by a grant from the NIA as well. So I'll take any questions now. Thank you.
I'll ask one.
All right.
I got the mic, I got the power.
Yeah.
So the wet-dry cycles you were showing where it was like raining off and then refilling and drying out again.
Yeah.
So you're thinking this is like a marginal coastal type environment where you're having some terrestrial exposure?
Yes, yeah.
Okay.
So our fundamental thing, you're looking at either small islands or very small proto-continents on
Sure,
there.
yeah.
And so we're not looking at bulk water systems at all. It is very hard to do chemistry in the bulk water.
Gotcha.
Behind you.
Any other?
I'm just curious, can this also work with ammonia oceans as well, or is it over just only considering just water oceans at this point?
So ammonia is a key component of it. So urea will convert to ammonia. Ammonia formate has ammonia in it. So you do need ammonia to drive these reactions. So ammonia oceans will work, ammonia lagoons will work, but you need some source of ammonia, yes.
In a prebiotic setting, could this phosphorylation be non-specific? Like you could end up phosphorylating your amino acids too? Or would this be somewhat specific at least?
So we have shown a small amount of regio-specific reactions. So like if you look at the adenosine, you form far more 5' than the 2' and the 3'. If you put in glycerol, it's going to react with that. I don't know how it's going to react with a whole mixture of alcohols in there and what will be specifically phosphorylated over there. It looks like it's a pretty non-specific reaction.
Okay.
How much water is too much water before the dehydration doesn't work very well anymore?
So, I mean, that's a good question. This is actually problematic to quantify in NMR. Because in this eutectic, you get so many interactions, we just get a broad peak instead of a nice, sharp water peak. It is what it is. But there is still water present in there. It takes 3 days of heating at 85 before it starts phosphorylate. If I re-add water, it stops, and then happens like a day to 2 days later. But I don't know how much is in there. But it is still in there.
Any other questions? All right, let's thank Bradley again.
Thank you.